Evolution of sociality in spiders, Stegodyphus, leads to depleted genomic diversity at both population and species levels. In this study, the effect of sociality on the genetic diversity of various spider species of the Stegodyphus genus in Africa was examined. Sociality involves cooperative breeding, female-based sex ratio, reproductive skew, and inbreeding. This often decreases effective population size, reducing the amount of genetic diversity via genetic drift.
Much lower levels of genetic diversity reduce the ability of a species to evolve over time in response to changes. How sociality reduces genetic diversity, and therefore evolutionary potential, is not well studied in spiders. The hypothesis being tested was whether sociality had an effect on the genetic diversity of the spiders.
232 spiders from 7 distinct species of genus Stegodyphus were extracted from various regions of Africa, and each individual's DNA was sequenced. The species of spiders sampled varied in sociality level, indicated by reproductive skew and inbreeding tendencies.
Estimates of genetic diversity were based on levels of observed individual heterozygosity within population diversity and species-wide diversity. Results of the experiment yielded much lower levels of genetic diversity in the social inbreeding species than the outcrossing subsocial species.
Diversity of species-wide nucleotides was 4 times lower in the inbreeding species than in the subsocial outcrossing ones, and within population diversity was 10 times lower. Average heterozygosity estimates of individuals were 6 times lower in the social species.
The results show that spider species with female bias, reproductive skew, and inbreeding have more limited diversity. Sociality also reduces effective population size, which will further enhance the negative effects of low diversity due to drift.
Low population may also limit recombination and selection, thus allowing deleterious characters to persist. The hypothesis that sociality among some species leads to limited genetic diversity and evolution was observed to be accurate.
This is detrimental to the spider species, as low diversity means it will be difficult to react to various changes or challenges that may occur in their environment.